Hey friends, I made it. I'm on Mount Everest. Woo! It's cold up here. And really, there's nobody here. I'm all by myself because they shut down the mountain. But I'm on Mount Everest. Woo! I can't, yeah. Woo! All right. Let me close it, shut this off. I can't see anything. Ugh. Woo! Air's pretty thin up here. I'm having a hard time breathing. Let me get all rid of that. All right. Woo! Look at this. This is so weird. Okay. I'm just going to show you. This is 10 hours of blizzard, snowstorm, Arctic howling wind sounds for sleeping, relaxing, and insomnia. Yeah, that's what I want to go to sleep to. Okay. So welcome to our meeting today. And I hope that you have all your notes and stuff in front of you. We are going to finish work. Oh, you guys can't see this. Oh, you can't see it because I'm not showing me. Okay. So, whoops, I got rid of me by accident. There we go. Okay. There, oh, it's kind of screwy. I have to, oh, it won't pick up the packet. So there we go. Okay, so if you have your packet, we're gonna go over that, but of course this is recorded. And so if you don't have it, then you can go back and finish filling this out. Yes, Destiny, you can, Desiree, you can come in and climb the mountain with me. I actually did work out this morning, so that was good. All right, so we're just, oh, hang on. They're mowing the lawn on Mount Everest as well. Okay, so I'm trying to decide where, so we left off starting in on our packet. So let's just go ahead and go to our packet. And then let me get rid of us. We don't need to see us. Okay, and then I want to annotate text. Blue's fine. Okay, that'll work. All right, so let's do this first. Let's finish this off. Maybe I'll, oh, here we go. Scroll right here. Oops. Okay, I'm going to get this from a different place because now my mouse isn't doing any. Uh-oh. Okay, sorry. Okay, I got something weird going on. I don't know how to get out of it, you guys. Oh my gosh. I don't even know what this is. And I can't escape either. I seriously have just lost how to do this. So let me get rid of that. Okay, now I know what I'm doing.
Oh my gosh, why is it taking so long to load?
All right. Okay. So here we are in controlled respiration. All right. So, all right. Okay, so let me go back and share this again. Okay. All right, now I know what I'm doing. It was my annotate thing that was messing me up. So I'm just gonna write in the words instead of typing them in. Okay, so what we need to take a look at then is how do we control respiration? We started talking about this. Let me pick a different color. before we left. And so this is the DRG. We'll see how well I can write with my mouse. Okay, so where in the medulla are the neurons that set the basic respiratory rhythm? A dorsal respiratory group. And it's going to be modified by the ventral respiratory group. And I'll show you a picture of that in just a second. Ventral respiratory group and the pons, or the pontine respiratory group. But I'm just going to write in pons right here. All right, now, we also discussed this a little bit before we left. What is the most important stimulus for controlling ventilation? And that's carbon dioxide. And the reason why is that in our brain, next to, and I'll show you a picture of it in just a second, next to the ventral or the dorsal respiratory group, there are central chemoreceptors. And they're central because they're in the central nervous system. And they are chemoreceptors for hydrogen ion. Okay, so hydrogen concentration, or not H2, sorry, it should be H plus. As I was writing that, I was like, wait a minute, what am I doing? Okay, oops. H plus, okay. Now, hydrogen ions can't cross the blood-brain barrier. And so the only way that you can change the pH of the cerebral spinal fluid is by changing the amount of CO2 that you have, because the CO2 can easily cross the blood-brain barrier because it's just a gas. But when it gets into the cerebral spinal fluid, then it's going to drop the pH, because remember this right here, we can never forget this. Carbon dioxide, oh gosh, plus water gives you carbonic acid and hydrogen ions and bicarbonate ions. Okay. Now, here's the next thing. Arterial PO2 must drop below wet to stimulate peripheral chemoreceptors. So in two places, oh, you know what? I'm going to go there right now because it'll be easy, and then we'll come back to this. So let me go to this. Okay. Okay. So, where's my, hang on. Okay. So here are my chemoreceptors. I have, hang on. Okay. So I have, let me pick a different color. Pick green. All right. So I've got two sets of chemoreceptors. I have peripheral chemoreceptors that are going to be here in the carotid sinus and in the aortic arch. And then I have the central chemoreceptors that are going to be right here in the medulla oblongata, right next to where the dorsal respiratory group is. Okay, now you can see that we have the 3 receptors for the different chemicals. So for a drop in O2, a rise in CO2, and a rise in hydrogen ions, which should be, because I don't like that, it's true, but remember, if you get more hydrogen ions, you're always going to get a drop in pH. Okay, so the question asked, why does the PO2 have to rise above or below 60 millimeters of mercury? So 60 before these suckers will get stimulated. Okay, so this will get stimulated quite easily. A rise in CO2, so my PCO2 levels being above 45 are going to be stimulated quite rapidly. My pH being lower than 7.35 will get stimulated quite rapidly, but I have to wait until my CO2 drops below 60, and here's why. Let's see. Oops, let me get rid of that. Okay, so let's go back to this graph right here. Yeah, this is the one I want. Okay, so here's what happens, why it has to drop below 60. Whoops. Okay, so if we look at a PO2 of 80 right here, you can still see we're about 95% saturated. So breathing harder isn't going to make any difference. I'm not going to load that much more oxygen onto my hemoglobin. Same with 70. I'm still at 90% saturation. Again, changing my breathing rate, because that's what we're talking about, is control of breathing. How fast and how deep are we breathing, or how slowly and shallowly are we breathing? But when we get to 60, right here, well, I guess there we're 90% saturated. But here's the deal. This is where the graph starts to slope down like this. And how often is arterial blood going to be at a PO2 of 60? Remember, arterial blood is 104. Okay, so I have to have a whole bunch of lung impairment to make it so that this blood is 60 millimeters of mercury, or I have to be holding my breath for a long time, or I have to be up at high elevations, or something that's going to make me breathe harder. Okay, so when it's 60 in here, that's when your peripheral chemoreceptors start going, wow, it's not oxygenated well at all here, so we better be breathing a lot harder to get in more oxygen. So you know, when I'm up there on Mount Everest, that I have to breathe really hard when I'm up there. And just imagine the fact that I'm climbing, that's going to be even more exertion. And so my oxygen demand is even going to be higher, and my body's going to have such a hard time being able to supply that oxygen. So I'm just going to be breathing super, super hard. All right, so let's go back to the packet. All right, oh, I erased all my drawings. Oh, well, that's okay. Okay, so we'll go back to this one. So arterial PO2 must drop below 60 to stimulate the peripheral chemoreceptors. Okay, so now if we go back to our formula again, and you know what? I'm gonna go ahead and type that in right there. And I'm just gonna use lines because I can't make arrows really quick. H2CO3, oops. And then H plus, oh, it won't let me subscript fast. And then HCO3 minus. Okay, so there's our formula. So if a person hyperventilates, what will happen to the following in the blood? Okay, so we know that in our tissues, we're going to proceed in this way. Okay, so this one we're picking up CO2. And then when we're in our lungs, we're going that way. Oops. Okay, and so which direction will we be going if we're hyperventilating? So we're breathing lots, so that means we're going to be taking a lot of this bicarbonate and hydrogen ions, converting it back to carbonic acid and becoming CO2 and water again so that we can breathe out the CO2. So when we're hyperventilating, I'm going to put hyper goes to the left, So when I'm breathing really hard and when I'm holding my breath, then the reaction is going to proceed to the right. Okay. Hypoventilating. All right. So if a person hyperventilates, what will happen to the following in the blood? Well, my CO2 levels will drop because I'm blowing off CO2, and that's an important thing to remember. When we hyperventilate, we blow off CO2, but because we're driving the reaction to the right, our pH is going to rise because we took away these hydrogen ions that were over here on the right. If we hypoventilate, then our pH drops. So because guess what? It's April next week. And so, yeah, we're almost but now we pushed everything back a week. So we're almost into acids and bases, friends. So when PCO2 drops, then pH rises. Okay, and then if a person hypoventilates, what will happen to the following in the blood? If I'm not breathing in, I'm not getting O2, and that means my tissues are going to be using it up. And so my PO2 levels will drop when I hypoventilate. And then if I'm hypoventilating, I'm building up CO2, so I'm going over here to the right, and so my PCO2 levels will increase. All right, so those are going to be the driving factors for breathing rate, which we'll get back into in just a minute. But then we have some other means of controlling respiration, and so they're these little reflex stuff right here. So the first one is that we have some stretch receptors in the lungs, okay, and oops. Stretch. Did I spell that right? Who knows anymore? I can't. Oh, okay. So stretch receptors in the lungs are going to be stimulated as our lungs expand. So this is going to be during inspiratory reserve volume. So when we're forcefully inhaling and we're using our neck muscles, our sternocleidomastoid, and our scalenes to bring our chest wall up and out, we don't want to overinflate the lungs. Okay, so no hyperinflation. And so there is a point at which you're filling up your lungs and like, oh, okay, I have to stop. I'm full of air. And so that will inhibit my dorsal respiratory group up here. And the effective respiration is to inhibit. So stretch receptors, when we're filling up our lungs, will only go so far. Okay, let me get this chat question. All right. I knew there was a T in there. Thanks, Trav. So I'm like, well, here was my thought process. I thought I already put the T in there. Oh, heaven's sakes. Okay. So stretch receptors, the effect is to inhibit. What is this reflex called? It's called the Herring, H-E-R. I think I can capitalize better than baby letters. Herring Breuer, B-R. It's German. E-U-E-R. Okay, all right, Herring Breuer reflex. All right, now I gotta click out of that. Okay, oops, there we go. So I'll take a screenshot of that or something because it's gonna go away right now. Okay, so clear my drawings. All right, so now we're gonna go to, okay, so there we go. And so now we wanna look at, okay, dust, smoke, and noxious fumes and coronavirus will stimulate receptors in the airway. Name the receptors. These are pulmonary irritants receptors, pulmonary irritant. And they will decrease our rate of breathing as well as stimulating the protective mechanisms down here of sneezing and coughing. So just make a little note to yourself. sneeze. Oh, love it. Oh, okay. Thanks. We're supposed to just have a 20-minute or a 40-minute time limit. Zoom said, hey, we took that away for you. So hooray. Okay, so sneeze and cough. Are the protective reflexes, and it makes total sense. I'm not going to write it in because it takes too long. You guys can write this down. Why do we sneeze and cough? Because we want to propel those substances out of our airway and give them to someone else. And so those are those protective reflexes. So what they'll do is they'll go up to, so we'll get those pulmonary irritants. Remember, because it's a reflex, the sensory input is going to go to the medulla. That's going to stimulate the dorsal respiratory group to contract, send signals to the phrenic nerve to fire harder so that we'll cough, or the muscles that are within our pharynx to produce a sneeze. All right. So the last one, and I'm going to go ahead and type these out because it'll be easier. Okay, so name 4 of the 6 factors that probably increase ventilation during exercise. Okay, so obviously we know a decrease in PO2. So decrease in PO2, okay? And actually it would be this one. This would be the first thing, increase in PCO2. And then because we have an increase in PCO2, we have a decrease in pH. Whoops. pH. And we also get, let's see, muscle stimulus. Okay. So even the contraction of our muscles can stimulate that respiratory group, that dorsal respiratory group, to fire more. So let's go to that in our lecture notes. So I'm going to go to the PowerPoint. Okay, share. All right, so we're going to go. Okay. So we did all of that. Okay, so now we're in right there. Okay, so carbon dioxide and blood pH. So changes in respiratory rate or depth produce changes in pH by altering bicarbonate or carbonic acid in the blood. Slow, shallow breathing causes a drop in pH, more carbonic acid, whereas, so we could call this, let me write on there. Hang on. Okay. Draw. Okay, so this right here is, oops, sorry about that. Oh, sorry about that. Oh, it's not letting me draw. Nope. Oh well, I won't write on it. So hypoventilation, and then rapid deep breathing flushes CO2 with a decrease in carbonic acid. So that's hyperventilation. I don't know why. Oh, it's letting me over here, but let me just check something really quick. All right. Okay. All right. So here's where all that takes place. So we already saw this. So here's the dorsal respiratory group right back here, and the ventral respiratory group in front, and the pontine respiratory groups that will influence that. And so here's when the dorsal respiratory group sends out its signals, then it's going to send signals here to the external intercostals and the diaphragm for inspiration, because it's going to govern inspiration. And then if we want to forcefully inhale, then we've got the signals that are being sent out, the stimuli being sent out to the internal intercostals. No, that's for forceful exhale, sorry. When we want to forcefully expire, then we'll be using the rest of our muscles to forcefully exhale. Okay, so this would be inhale, this would be exhale. All right. As I said before in class, that the dorsal respiratory group is kind of like the SA node in that it will fire spontaneously, so depolarize spontaneously about 12 to 15 times per minute, because that's our homeostasis. That's about how fast we have to breathe in order to get the CO2 out of the system and the O2 to our muscles as they're respiring. But like you know, if you're exercising, you have more O2 demand. You also are producing more CO2 that you need to get rid of. And so those, and the drop in pH that will accompany that. And so those things will affect your breathing rate. That will make you breathe faster, and it will make you breathe deeper. Okay, so the ventral respiratory group, active during forced ventilation. So we saw that when it was going to the ventral respiratory group, receives the input from the DRG for forced inspiration and expiration during stress and exercise. And then the interaction between the DRG and DRG begins with slow signals and gradually increases to produce a smooth inspiratory effort instead of a gasp. So we're not just all the time. Okay, then the pons has two centers in it, the apneustic and pneumotaxic. It's going to get the information, so the sensory input that's going to come into the dorsal respiratory, well, come into the medulla and then send signals down to the dorsal respiratory group to adjust. So the apneustic center stimulates the inspiratory neurons of the DRG and the VRG, and then the pneumotaxic sends inhibitory centers to the inhibitory signals to the inspiratory centers. Those are in the PowerPoint that's in Canvas. Yep. So all of this is the same PowerPoint that because Marcus just said, are those slides shared with us in Canvas? Yeah, everything that I have is what you guys have. And so, yeah, if you go to the slides in Canvas, they're there to my PowerPoints. All right. Okay, and then PRG, or pontine respiratory group, helps adjust breathing rate and patterns for sleep and exercise. Okay. So the idea is that we have two neuron networks in the dorsal respiratory group. One that's our start neurons, and our start neurons fire for about 2 seconds, and so then they send stimulatory signals to the diaphragm through the phrenic nerve, and so we contract for 2 seconds, and then there's a set of stop neurons that fires and inhibits the start ones. And by inhibiting the start ones, then the signals are not being sent to the phrenic nerve anymore. Let me see if I can make that, because I want to draw that picture, and I don't know why it's not letting me annotate over here. I don't know. Let me stop sharing and let me share again. Well, let me do that. Okay, now let me do this. Okay. Oh, there we go. Okay, so here we go. This is how it's going to work. So I have my start neurons. Oh, you know what? This annoys me. Start and stop neurons. These are both in the DRG. Okay, so stop. DRG. So these neurons we're going to fire. I'm going to type this. Okay. So they fire for 2 seconds. Okay, then that sends signal to phrenic nerve. And then diaphragm. Contracts. Okay, so that's going to be inspiration. Okay, so then our stop neurons are for expiration. So then they're going to fire for 3 seconds, and then no signal to phrenic nerve, and diaphragm relaxes, And then we get, all right, now think about that. That means the whole respiratory cycle. Okay, so we'll put 1 respiratory cycle equals 5 seconds. Okay, so then if we're breathing, if it takes 5 seconds to do 1 breath, then how many times can we breathe in a minute? Oh, look at that. And that's what we said. That is our intrinsic rate of breathing. Okay, so now if we're going to change our breathing rate, then we're going to infringe on this stop time. So we're going to decrease the time that we're not breathing. And so that will make this, will decrease this. And you know, guys, we're never going to get to a breathing rate of about 20, more than 20, because if we're breathing faster than 20 times a minute, then we blow off too much CO2, and then there's no stimulus to the brain, and then we get apnea. And so we need to have CO2 or else we don't breathe. And so if you want to hold your breath for a really long time, the best thing to do is hyperventilate before you try to hold your breath, because you're blowing off lots of CO2, and then you take one last breath and you hold that, and then you have to wait for your CO2 levels to build back up again before your brain will want to inhale again. So so that's just something there. So anyway, so that's how all of that works. All right, let's move on. Oops. All right, so here are the chemical factors then again that are going to control respiration. So CO2, most potent in influencing respiration. If we get too much CO2, we get hypercapnia, and then CO2 in the cerebrospinal fluid from the blood decreases pH through the same reaction occurs in the red blood cells. Central chemoreceptors in the ventrolateral medulla stimulated to increase breathing rate. Whoops. Rising CO2 levels change breathing rate is the regulation of hydrogen ions to the brain that truly drives respiration rates. Even though CO2 is our stimulus for breathing, it's the pH of our cerebrospinal fluid that's making us respire. But you can't get a change in pH in your cerebrospinal fluid unless you have a change in CO2. So then in summary, a drop in pH and a rise in CO2 stimulate rapid breathing. When CO2 levels are abnormally low, apnea may occur. Okay, so that's what I just told you about. And then high pH and low CO2 stimulate breath holding. So I want you to think about when you're asleep. Okay, how much CO2 are you actually producing? Hey, not very much. So what is the pH of your blood going to be? More high than normal. And so that's why we don't breathe as much while we sleep. And again, we'll talk more about that when we get into acid-base regulation. Okay, so then we've got, so that was central chemoreceptors. These are the peripheral chemoreceptors. And so they're going to be the ones that are in the arch of the aorta and the carotid sinus. So they're going to detect rising CO2. They're also going to detect a PO2 but less than 60. And then a drop in pH and exercise increases ventilation due to both the combined use of O2 and generation of CO2. And when we're at high altitudes, primarily, so when I was up there on Everest this morning, primarily the lack of O2, because our PO2 is so low at high elevations, that's what's driving our respiration and not too much CO2. So there's that video again, that slide again, showing peripheral chemoreceptors, and then we've got receptors in muscles and joints that are saying, hey, look, we're exercising, you might want to start breathing again, or breathing faster and deeper because we're going to need some O2 in a minute. Here, the pulmonary irritant receptors in the trachea and the bronchi of the lungs, notice that they have an inhibitory effect here on the respiratory centers. Same with the stretch receptors in the lungs. They're going to be having an inhibitory effect as well. These will all have stimulatory effects so that you'll breathe faster and deeper. This is slower and more shallowly. And then we have sympathetic, parasympathetic input here that will affect the hypothalamus and then either increase or decrease our breathing rate. And then higher brain centers, of course, we can breathe faster or slower depending on what we want to do. And so, so it depends on what's going on, whether it's a stimulatory effect or an inhibitory effect. Pain and fear are kind of cool. I mean, they're not, but they're opposite. So, which is strange because they're both coming, you know, it's both sympathetic and autonomic. And so when we think of pain, like if you slam your finger in the door, what are you going to do? You hold your breath, right? Little kids. Oh, I'll get to that in a minute. So yeah, pain usually makes us hold our breath. Okay, so think about it. When you're exercising, people out there, what do they always tell you? Don't hold your breath. Keep breathing. Whenever I'm doing abs, I always want to hold my breath. I don't like to breathe while I'm because it hurts. And so I want to hold my breath. But when we're afraid of something, what do? Oh, we hyperventilate. We're just like, you know, freak out and have a hissy fit or something like that. I don't know. But anyway, so it just depends what pain and emotion are going to do to us. It's going to vary. Now, here's a cool thing I want you to think about. I don't know how many of you have ever witnessed this or know that this is a thing, but sometimes there are babies and little kids that will pass out when they cry. So what's going on? Well, they're crying. They're blowing off CO2. So they're driving the reaction to the left. Lots of CO2 is leaving. And so what does that do? No CO2, no rise in CO2, no stimulus for the medulla, and they'll actually stop breathing and pass out. So it can be scary if you don't know that that's a thing, but it's not that big a deal. They'll start breathing again. And also you can, you know, if you've got a niece or a nephew or a child or somebody that says, I'm gonna hold my breath till I die. Hey, do it, okay? Call them on it and say, go right ahead, because guess what? You're not going to, because as you hold your breath, your CO2 levels will rise and you will have an uncontrollable urge to breathe again when your peripheral, well, it's mostly your central chemoreceptors in your brain are going, I have had enough of this crap. Start breathing again. And you will.
So they'll always start breathing,
They'll
Miss G?
always start breathing unless you have some other underlying pathology, which, you know, 99 times out of 100, no, they don't. But yeah, they'll always start breathing again.
My niece did that last week and turned like completely purple, and I'm like, turn her around and make her breathe, because she was screaming.
Yeah, but she'll breathe again. Yeah, so yeah, it can be scary, but know that, yeah, nature will take its course. I know when Amy had a seizure, I think I already told you about this, that it blocks her ability to breathe, and so she turned totally, the whites of her eyes were blue, her sclera was blue, and her lips were blue, but yeah, she started breathing again. So after the seizure was over, so it was all done. So that's that. Hang on. Just a sec. I got to text somebody. Okay. So we did pulmonary irritants. There it is. Coughing and sneezing. Herring-Breuer. There's that is. And then hypothalamic control. Emotions, pain can increase or decrease. And then whatever. So we're under voluntary control, except when CO2 levels are too high and the brainstem reinitiates breathing. All right, and then here's that. You can look at, there's arterial PCO2 and what everything's gonna do in our chemoreceptors, and we already went through that, and there's all of that, and there's all that. There's all that, there's all that. You can look at all that. I don't even know what this video is. So you can click on that and watch it. I don't even know what it is anymore, but I'm not gonna waste our time in doing that. Okay, So, oh, that was good. That was only 40 minutes. All right. So, I want to start in on some digestion stuff right now. So, I'm going to take the next 10 minutes and then, because we're still going to meet on Friday, and then we'll go ahead and really start into digestion and with vengeance on Friday. But since we're here and we still have at least 10 more minutes left in our regular class, and then we'll do the lab stuff, and the lab stuff's not very long, so so that'll be good. Okay, so can you guys see? Somebody just, you know, chat me and say yes, we can see digestive system. I don't know if I need to stop sharing and reshare, or somebody get on and say yes, I can see it. Whoops. Okay, sweet. All right, thank you very much. Because I have no clue. I don't know what I can see, and I mean what you see and what or not. Okay, so anyway, okay, what are the objectives of the digestive system? Okay, there's that. You know how we usually have a page of objectives? You know, 7, not so bad. Ha! Look at that. We have 14 objectives in the digestive system. But I really spelled it out, so it's not that bad. Okay, so major processes occurring during digestive system activity. Boom, there they are. So let's look at the picture first, and then we'll go back to the words. So here is what we do when we eat. So the very first thing is that we ingest our food. We take it in through our mouth, and it goes into our pharynx. While it's in the mouth, we have 2 things going on. We have mechanical digestion. So we have chewing, obviously, to help mechanically break down the food. But we also have a little bit of chemical digestion. If you remember the chart that we did in lab that had salivary amylase that begins the chemical digestion of carbohydrates. Then the food will go down through our pharynx till it gets to the esophagus, and then we go through propulsion. So ingestion, deglutition is swallowing, and then we need to move the food along. So we'll have muscle contraction, and it's going to be voluntary. The top 2/3 of the how's it, top 2/3, top third portion, I'll just go with the portion of the esophagus is still skeletal muscle. Once we get from pharynx into esophagus, and then we transition down into smooth muscle, so that we have peristalsis happening. So peristalsis is that rhythmic contraction of the smooth muscle. And remember how we talked about smooth muscle contracts like wringing out a towel. And so it's going to squeeze around the lumen of the esophagus and the rest of the alimentary canal, and then push the food along. So you can see peristalsis happens in all 4 places: esophagus, stomach, small and large intestine. Okay, then we have there should have been an arrow of chemical digestion up there, but whatevs. Okay, so then we get into the stomach, and so we're still propelling, but here's where we're going to be adding the chemical or the hydrolytic enzymes to break up the food into absorbable pieces. And then when we get to the small intestine, then we will absorb the food. And then as we pass through this to large intestine, we still have absorption going on. And we'll talk about this difference into a blood vessel or a lymph vessel. And then notice that large intestine absorption is mainly water. And then once we've taken out everything that we can take out, then it becomes our feces, and then defecation will propel that or move that material out through the anus. Okay, so there's those processes: ingestion, propulsion, mechanical. Oh, churning. Let's go back. Churning is when your stomach moves back and forth, and we'll talk about how your stomach has the ability to churn here in just a minute, or maybe Friday. And then segmentation is the other thing. And I already talked to you about segmentation when we were in lab, but I'll tell you about it here again in just a minute. And then chemical digestion, absorption, defecation. All right, so let's take a look at these 2 mechanical processes that are going on. We have peristalsis. So notice that we get smooth muscle contraction behind the bolus of food, for example, whatever we're trying to move. And so it's going to be like squeezing toothpaste in the proper way of squeezing toothpaste out of the toothpaste tube. Oh my gosh, I hate toothpaste being squeezed in the middle of the tube. Drives me crazy. Because it's this. When you squeeze the toothpaste in the middle of the tube, you don't get peristalsis, you get segmentation. So here's what happens first. So I get contraction in little areas along the smooth muscle, along the small intestine. So here I have little blobs of food. And now what I'm going to do is squeeze in the middle of the segment, okay, segmentation. And look at what happens when I squeeze in the middle of a segment. Half of the material goes up to the previous segment, half of it goes to the lower, or the superior segment, and this goes to the inferior lower segment. And then I squeeze in the middle of a segment again, back to my original position. And oh, look at what I did. I mixed the food back and forth. And so what you're doing is just squeezing up and down and up and down and up and down and up and down in order to get as much exposure of the food to the hydrolytic enzymes so that chemical digestion can occur. So this is segmentation at the bottom, and this is what happens in the small intestine. Okay, let me go through the layers of the digestive system, and then we'll take like a 5-minute break, and then we'll come back and do lab. So here is, let's look at this picture first. All right, so if we cut through any place in the digestive system, essentially from the stomach to the rectum, esophagus is going to be a little bit different than this, but absolutely, well, I guess we could include this part of the esophagus all the way down to the rectum. It's going to look just like this, no matter what the diameter of the tube is, whether it's small intestine or large intestine. This is what we're going to see. So we're going to start with the inner layer of the alimentary canal, or the food canal. That's what alimentary canal means. So we're going to go to the mucosa. So here is the inner layer of our digestive system. And of course, we're going to have an epithelial layer. Remember, you guys, that this is, let me get my annotate. Okay, that this is now simple columnar epithelium. Okay, and we also need to have some goblet cells. Okay, that are going to be interspersed in this lining right here. Okay, then we have what's known as a lamina propria. So it's kind of like basement membrane, but thicker, a little more complex. We've got some glands in here, some crypts of Lieberkühn. Oh, I love that name. With secretions that are going to occur from there. And then we also have just this one, they make it look like it's 2 cell layers thick. It's about 1 cell layer thick of some smooth muscle, but we don't really need to worry about that. So, but it's there. Okay, so that is our mucosa, simple columnar epithelium with goblet cells, and our lamina propria, and just our little muscularis. Okay, then we're going to come to the submucosa, which is primarily going to be areolar. Oops, I put the O in the wrong place. No, I spelled that whole thing wrong. There we go. Okay. There we go. All right, and we're going to have also the glandular material in here. This is important right here. Our mucosa-associated lymphoid tissue, Peyer's. Remember our Peyer's patches. Okay, so this is going to be clusters of lymphoid tissue. It's going to be like a lymph node, but not capsulized. So it doesn't have a capsule around. It's not a real organized organ per se, or gland per se, node, but it is clusters of lymphoid tissue. And so, so that's the MALT, mucosa-associated lymphoid tissue. Okay, and then oh, I forgot that. That's in the sorry, that's in the mucosa, not in the submucosa. Okay, but remember, there's the mold. Okay, so then we have submucosa. We've got blood vessels that are going to nourish the cells that are inside of here, and we have nerve endings. Hey, guess what? We've got 2 nerve plexuses. I don't know if you're familiar with your enteric nervous system. So we have a central nervous system, we have a peripheral nervous system, but then we also have an enteric nervous system. And our enteric nervous system is like our gut brain, and it really fires independently of our brain. Is that we'll get sensory input that goes into the enteric nervous system and then comes back out. So we don't have to go even all the way to the spinal cord to get reflexes to happen. So we've got 2. We've got an underlying and innermost one called the submucosa nerve plexus. And so these are going to be, oh, look, there's some ganglia. So we know we've got some cell bodies in there that are going to go to the submucosa. And then we have the myenteric nerve plexus, which is going to be a set of nerves and ganglia that are going to go to the next layer of the digestive tract, which is the muscularis, or muscularis externa. Now we've got 2 layers of muscular tissue. In smooth muscle, we have a longitudinal layer, so the cells are all running this way, so that when we look at them in cross section, we just see the cells end on like that. And then we have circular, so the cells are going around. So if we cut through, we can see cells end on this way, but now we're seeing entire cells as they run around in a circle. So we have longitudinal, which is going to help with peristalsis, obviously, and then the circular layer, which is going to increase and decrease the diameter of the lumen. And then on the outside is the serosa, okay, or serous membrane. So this is going to be, you guys should tell me, visceral peritoneum. Okay, all right, so that is all that we're going to do. Let me just get rid of my drawings and go back to, whoops, oh, get rid of that, and then go back to this. Whoop, yeah, okay, so, oh yeah, sorry about that. Okay, so the mucosa or mucous membrane, secretion, absorption, protection. They have simple columnar epithelium, dense, well, I put loose, between a relar and dense irregular connective tissue. So blood, lymphatic vessels, lymph nodules, and nerves, muscularis, and inner circular layer, outer longitudinal layer, and then the serosa is the visceral peritoneum, a relar connective tissue with simple squamous epithelium on the outside. Okay, so that's where we're going to stop for the actual lecture video, and I'm going to